My talk is going to focus on three aspects. First of all, I'm going to show how we use fly genetics plus medicinal chemistry plus computational chemistry to build drugs step by step that hit multiple targets. The complexity that we build into the drug is designed to deal with the complexity of the tumor. That's the first topic. And we're even using that approach to take currently approved drugs and create what we call super versions of them that correct their deficiencies. The second thing I'm going to talk about is how we build complex fly models. We can build up to 12 or 15 hit fly models that are designed to capture the genetic complexity of individual cancer patients. And what I'm going to show is that as the complexity of the model goes up, the number of drugs that will work against it goes down, and that you really have to go to cocktails of drugs or polypharmacology, which has really been motivating our drug design. And then I'm going to finish by talking about our efforts at the Center for Personalized Cancer Therapeutics, which I opened relatively recently. We currently have 25 patients in the center, and we sequence their tumors. We build personalized fly avatars. We do high-throughput drug screening using robotics to create recommendations for treatments to the patient and their oncologist. Yeah, what we do is a little unusual in that sense. There has been a debate in the field about what's called targeted therapy, which really means, has come to mean hitting a single target with the idea that the cleaner you hit it, the less toxic the drug will be. That hasn't always panned out. Also, issues of resistance, tumor heterogeneity, that's had some difficulties. It's had successes, but also difficulties. The problem with polypharmacology, I don't think most people have a conceptual problem with it. The problem with it has always been that if you create what we would call a dirty drug that hits many targets, which we like, chemists don't really know what to do with it. So if you want to take a drug, for example, from our flies and improve it so it has better druggable properties to a patient, and you don't know all the activities that are important, you don't know what you can mess with and what you can't. So that motivated us to create a pipeline where we actually develop the drug stepwise. We know all the targets that matter. In fact, we've engineered some of them in. And then we can go to the chemist and say, yeah, you can get ADMET properties, PK, PD, all that stuff that you need to optimize it to humans, but don't touch these activities. And here's the assays for monitoring them. So we're basically giving this type of approach, polypharmacology, a rational way forward. Also, what we find when we do the screening in whole animal system, it's not just that we find drugs that hit many targets. We find, using our fly genetics, we can show that many of those targets are actually not required in the tumor. They're required in the microenvironment, they're required distantly in the skeletal muscle, in the fly liver, and so on, that then feed back to help choke the tumor out. Those sorts of activities you can't capture those in what I would call tumor-focused screening. And they're very difficult to keep track of for a chemist. This is why it's important for us to be able to map all of these things out and explain to the chemist what they can and can't do to the drug. So for those reasons, we've had to build that structure to allow us to take that approach. Well, we did help identify vandetanib, which has now been approved as standard of care for medullary thyroid carcinoma. We have developed other drugs which we've published that have now been licensed by biotech companies with the intent of bringing them into clinical trials. And as I mentioned, we've also used a modified version of this approach to consider treating patients. Well, first of all, one of the beauties of working in flies is that they're very cheap. So I don't envy my mouse colleagues who have mouse costs, which are pretty insane. So actually cost is mainly manpower. We do get funding both from the US government, from Mount Sinai, who has provided funding for our center, and of course from private foundations. So people are curious about this approach, and so and people have been willing to basically give us some room to play in this arena. First of all, I have to say I stole the name Avatar from Pier Paolo Pandolfi, who's made mouse avatars. with a somewhat similar intent. So we sequence the patient's tumor, and then the difficult part comes. So the patient will have mutations in many genes. And so we'll sit down with Eric Schott's bioinformatics group and go through them gene by gene and figure out which ones are actually altering protein function. That will generally bring the number down to between 5 and 15. If we can't bring it down further than that, then we have access to the Fly genetic tools. We'll actually test the candidates against what we call a base model and functionally figure out which ones matter. The key point is, at the end of the day, we're not looking for what's common between patients. Oh, I recognize this as a driver of cancer, therefore I will add that to the model. We try to take everything that we have evidence is functional. And if we're not sure, it goes in.